Chapter 11 Meiosis and Sexual Reproduction Figure 11.1 Each of us, like the organism shown above, begins life as a fertilized egg, zygote. After trillions of cell divisions, each of us develops into a complex, multicellular organism. Credit A. Modification of work by Frank Wouters. Credit B. Modification of work by Ken Cole, USGS. Credit C. Modification of work by Martin Pettit. Introduction. The ability to reproduce is a basic characteristic of all organisms. Hippopotamuses give birth to hippopotamus calves. Joshua trees produce seeds from which Joshua tree seedlings emerge. And adult flamingos lay eggs that hatch into flamingo chicks. However, unlike the organism shown above, offspring may or may not resemble their parents. For example, in the case of most insects such as butterflies, with a complete metamorphosis, the larval forms rarely resemble the adult forms. Although many unicellular organisms and a few multicellular organisms can produce genetically identical clones of themselves through asexual reproduction, many single-celled organisms and most multicellular organisms reproduce regularly using another method, sexual reproduction. This highly evolved method involves the production by parents of two haploid cells and the fusion of two haploid cells to form a single, genetically recombined diploid cell, a genetically unique organism. Haploid cells that are part of the sexual reproductive cycle are produced by a type of cell division called meiosis. Sexual reproduction, involving both meiosis and fertilization, introduces variation into offspring that may account for the evolutionary success of sexual reproduction. The vast majority of eukaryotic organisms, both multicellular and unicellular, can or must employ some form of meiosis and fertilization to reproduce. In most plants and animals, through thousands of rounds of meiotic cell division, diploid cells, whether produced by asexual or sexual reproduction, will develop into an adult organism. Chapter Outline 11.1 The process of meiosis 11.2 Sexual reproduction 11.1 The process of meiosis. By the end of this section, you will be able to do the following. Describe the behavior of chromosomes during meiosis, and the differences between the first and second meiotic divisions. Describe the cellular events that take place during meiosis. Explain the differences between meiosis and mitosis. Explain the mechanisms within the meiotic process that produce genetic variation among the haploid gametes. Sexual reproduction requires the union of two specialized cells, called gametes, each of which contains one set of chromosomes. When gametes unite, they form a zygote, or fertilized egg that contains two sets of chromosomes. Note: cells that contain one set of chromosomes are called haploid. Cells containing two sets of chromosomes are called diploid. If the reproductive cycle is to continue for any sexually reproducing species, then the diploid cell must somehow reduce its number of chromosome sets to produce haploid gametes. Otherwise, the number of chromosome sets will double with every future round of fertilization. Therefore, sexual reproduction requires a nuclear division that reduces the number of chromosome sets by half. Most animals and plants and many unicellular organisms are diploid and therefore have two sets of chromosomes. In each somatic cell of the organism, all cells of a multicellular organism except the gametes or reproductive cells, the nucleus contains two copies of each chromosome, called homologous chromosomes. Homologous chromosomes are matched pairs containing the same genes in identical locations along their lengths. Diploid organisms inherit one copy of each homologous chromosome from each parent. Meiosis is the nuclear division that forms haploid cells from diploid cells, and it employs many of the same cellular mechanisms as mitosis. However, as you have learned, mitosis produces daughter cells whose nuclei are genetically identical to the original parent nucleus. In mitosis, both the parent and the daughter nuclei are at the same, ploidy level, diploid in the case of most multicellular animals. Plants use mitosis to grow as sporophytes, and to grow and produce eggs and sperm as gametophytes, so they use mitosis for both haploid and diploid cells, as well as for all other ploidies. In meiosis, the starting nucleus is always diploid and the daughter nuclei that result are haploid. To achieve this reduction in chromosome number, meiosis consists of one round of chromosome replication followed by two rounds of nuclear division. Because the events that occur during each of the division stages are analogous to the events of mitosis, the same stage names are assigned. However, because there are two rounds of division, the major process and the stages are designated with a I, or a 2. Thus, meiosis I is the first round of meiotic division and consists of prophase I, prometaphase I, and so on. Likewise, meiosis II, during which the second round of meiotic division takes place, includes prophase II, prometaphase II, and so on. Meiosis I. Meiosis is preceded by an interphase consisting of G1, S, and G2 phases, which are nearly identical to the phases preceding mitosis. The G1 phase, the first gap phase, is focused on cell growth. During the S phase, the second phase of interphase, the cell copies or replicates the DNA of the chromosomes. Finally, in the G2 phase, the second gap phase, the cell undergoes the final preparations for meiosis. During DNA duplication in the S phase, each chromosome is replicated to produce two identical copies, sister chromatids that are held together at the centromere by cohesin proteins, which hold the chromatids together until anaphase II. Prophase I. Early in prophase I, before the chromosomes can be seen clearly with a microscope, the homologous chromosomes are attached at their tips to the nuclear envelope by proteins. As the nuclear envelope begins to break down, the proteins associated with homologous chromosomes bring the pair closer together. Recall that in mitosis, homologous chromosomes do not pair together. The synaptonemal complex, a lattice of proteins between the homologous chromosomes, first forms at specific locations and then spreads outward. 280 Chapter 11. Meiosis and Sexual Reproduction. Access for free at openstax.org. To cover the entire length of the chromosomes. The tight pairing of the homologous chromosomes is called synapsis. In synapsis, the genes on the chromatids of the homologous chromosomes are aligned precisely with each other. The synaptonemal complex supports the exchange of chromosomal segments between homologous non-sister chromatids, a process called crossing over. Crossing over can be observed visually after the exchange as chiasmata, singular equals chasma, figure 11.2. In humans, even though the X and Y sex chromosomes are not completely homologous, that is, most of their genes differ, there is a small region of homology that allows the X and Y chromosomes to pair up during prophase I. A partial synaptonemal complex develops only between the regions of homology. Figure 11.2. Early in prophase I, homologous chromosomes come together to form a synapse. The chromosomes are bound tightly together and in perfect alignment by a protein lattice called a synaptonemal complex and by cohesin proteins at the centromere. Located at intervals along the synaptonemal complex are large protein assemblies called recombination nodules. These assemblies mark the points of later chiasmata and mediate the multi-step process of crossover, or genetic recombination, between the non-sister chromatids. Near the recombination nodule, the double-stranded DNA of each chromatid is cleaved, the cut ends are modified, and a new connection is made between the non-sister chromatids. As prophase I progresses, the synaptonemal complex begins to break down and the chromosomes begin to condense. When the synaptonemal complex is gone, the homologous chromosomes remain attached to each other at the centromere and at chiasmata. The chiasmata remain until anaphase I. The number of chiasmata varies according to the species and the length of the chromosome. There must be at least one chasma per chromosome for proper separation of homologous chromosomes during meiosis I, but there may be as many as 25. Following crossover, the synaptonemal complex breaks down and the cohesin connection between homologous pairs is removed. At the end of prophase I, the pairs are held together only at the chiasmata, figure 11.3. These pairs are called tetrads because the four sister chromatids of each pair of homologous chromosomes are now visible. The crossover events are the first source of genetic variation in the nuclei produced by meiosis. A single crossover event between homologous non-sister chromatids leads to a reciprocal exchange of equivalent DNA between a maternal chromosome and a paternal chromosome. When a recombinant sister chromatid is moved into a gamete cell, it will carry some DNA from one parent and some DNA from the other parent. The recombinant chromatid has a combination of maternal and paternal genes that did not exist before the crossover. Crossover events can occur almost anywhere along the length of the synapsed chromosomes. Different cells undergoing meiosis will therefore produce different recombinant chromatids, with varying combinations of maternal and parental genes. Multiple crossovers in an arm of the chromosome have the same effect, exchanging segments of DNA to produce genetically recombined chromosomes. 11.1. The process of meiosis. 281. Figure 11.3 crossover occurs between non-sister chromatids of homologous chromosomes. The result is an exchange of genetic material between homologous chromosomes. Prometaphase I. The key event in prometaphase I is the attachment of the spindle fiber microtubules to the kinetochore proteins at the centromeres. Kinetochore proteins are multiprotein complexes that bind the centromeres of a chromosome to the microtubules of the meiotic spindle. Microtubules grow from microtubule organizing centers, MTOCs. In animal cells, MTOCs are centrosomes located at opposite poles of the cell. The microtubules from each pole move toward the middle of the cell and attach to one of the kinetochores of the two fused homologous chromosomes. Each member of the homologous pair attaches to a microtubule extending from opposite poles of the cell so that in the next phase, the microtubules can pull the homologous pair apart. A spindle fiber that has attached to a kinetochore is called a kinetochore microtubule. At the end of prometaphase I, each tetrad is attached to microtubules from both poles, with one homologous chromosome facing each pole. The homologous chromosomes are still held together at the chiasmata. In addition, the nuclear membrane has broken down entirely. Metaphase I. During metaphase I, the homologous chromosomes are arranged at the metaphase plate, roughly in the midline of the cell, with the kinetochores facing opposite poles. The homologous pairs orient themselves randomly at the equator. For example, if the two homologous members of chromosome 1 are labeled A and B, then the chromosomes could line up AB or BA. This is important in determining the genes carried by a gamete, as each will only receive one of the two homologous chromosomes. Recall that after crossing over takes place, homologous chromosomes are not identical. They contain slight differences in their genetic information, causing each gamete to have a unique genetic makeup. The randomness in the alignment of recombined chromosomes at the metaphase plate, coupled with the crossing over events between non-sister chromatids, are responsible for much of the genetic variation in the offspring. To clarify this further, remember that the homologous chromosomes of a sexually reproducing organism are originally inherited as two separate sets. 282 Chapter 11. Meiosis and Sexual Reproduction. Access for free at openstax.org. One from each parent. Using humans as an example, one set of 23 chromosomes is present in the egg donated by the mother. The father provides the other set of 23 chromosomes in the sperm that fertilizes the egg. Every cell of the multicellular offspring has copies of the original two sets of homologous chromosomes. In prophase I of meiosis, the homologous chromosomes form the tetrads. In metaphase I, these pairs line up at the midway point between the two poles of the cell to form the metaphase plate. Because there is an equal chance that a microtubule fiber will encounter a maternally or paternally inherited chromosome, the arrangement of the tetrads at the metaphase plate is random. Thus, any maternally inherited chromosome may face either pole. Likewise, any paternally inherited chromosome may also face either pole. The orientation of each tetrad is independent of the orientation of the other 22 tetrads. This event, the random, or independent, assortment of homologous chromosomes at the metaphase plate, is the second mechanism that introduces variation into the gametes or spores. In each cell that undergoes meiosis, the arrangement of the tetrads is different. The number of variations is dependent on the number of chromosomes making up a set. There are two possibilities for orientation at the metaphase plate. The possible number of alignments therefore equals 2n in a diploid cell, where n is the number of chromosomes per haploid set. Humans have 23 chromosome pairs, which results in over 8,223,000 possible genetically distinct gametes just from the random alignment of chromosomes at the metaphase plate. This number does not include the variability that was previously produced by crossing over between the non-sister chromatids. Given these two mechanisms, it is highly unlikely that any two haploid cells resulting from meiosis will have the same genetic composition. Figure 11.4. To summarize, meiosis I creates genetically diverse gametes in two ways. First, during prophase I, crossover events between the non-sister chromatids of each homologous pair of chromosomes generate recombinant chromatids with new combinations of maternal and paternal genes. Second, the random assortment of tetrads on the metaphase plate produces unique combinations of maternal and paternal chromosomes that will make their way into the gametes. Figure 11.4 Random, independent assortment during metaphase I can be demonstrated by considering a cell with a set of 211.1. The process of meiosis 283 chromosomes, n equals 2. In this case, there are two possible arrangements at the equatorial plane in metaphase I. The total possible number of different gametes is 2n, where n equals the number of chromosomes in a set. In this example, there are 4 possible genetic combinations for the gametes. With n equals 23 in human cells, there are over 8 million possible combinations of paternal and maternal chromosomes. Anaphase I. In anaphase I, the microtubules pull the linked chromosomes apart. The sister chromatids remain tightly bound together at the centromere. The chiasmata are broken in anaphase I as the microtubules attached to the fused kinetochores pull the homologous chromosomes apart. Figure 11.5. Telophase I and cytokinesis. In telophase, the separated chromosomes arrive at opposite poles. The remainder of the typical telophase events may or may not occur, depending on the species. In some organisms, the chromosomes, decondense, and nuclear envelopes form around the separated sets of chromatids produced during telophase I. In other organisms, cytokinesis, the physical separation of the cytoplasmic components into two daughter cells, occurs without reformation of the nuclei. In nearly all species of animals and some fungi, cytokinesis separates the cell contents via a cleavage furrow, constriction of the actin ring that leads to cytoplasmic division. In plants, a cell plate is formed during cell cytokinesis by Golgi vesicles fusing at the metaphase plate. This cell plate will ultimately lead to the formation of cell walls that separate the two daughter cells. Two haploid cells are the result of the first meiotic division of a diploid cell. The cells are haploid because at each pole, there is just one of each pair of the homologous chromosomes. Therefore, only one full set of the chromosomes is present. This is why the cells are considered haploid. There is only one chromosome set, even though each chromosome still consists of two sister chromatids. Recall that sister chromatids are merely duplicates of one of the two homologous chromosomes, except for changes that occurred during crossing over. In meiosis II, these two sister chromatids will separate, creating 4 haploid daughter cells. Link to learning. Review the process of meiosis, observing how chromosomes align and migrate, at meiosis, and interactive animation. http://openstax.org/l/animal_meiosis. Meiosis II. In some species, cells enter a brief interphase, or interkinesis, before entering meiosis II. Interkinesis lacks an S phase, so chromosomes are not duplicated. The two cells produced in meiosis I go through the events of meiosis II in synchrony. During meiosis II, the sister chromatids within the two daughter cells separate, forming 4 new haploid gametes. The mechanics of meiosis II are similar to mitosis, except that each dividing cell has only one set of homologous chromosomes, each with 2 chromatids. Therefore, each cell has half the number of sister chromatids to separate out as a diploid cell undergoing mitosis. In terms of chromosomal content, cells at the start of meiosis II are similar to haploid cells in G2, preparing to undergo mitosis. Prophase II. If the chromosomes decondensed in telophase I, they condense again. If nuclear envelopes were formed, they fragment into vesicles. The mTOCs that were duplicated during interkinesis move away from each other toward opposite poles, and new spindles are formed. Prometaphase II. The nuclear envelopes are completely broken down, and the spindle is fully formed. Each sister chromatid forms an individual kinetochore that attaches to microtubules from opposite poles. Metaphase II. The sister chromatids are maximally condensed and aligned at the equator of the cell. Anaphase II. The sister chromatids are pulled apart by the kinetochore microtubules and move toward opposite poles. Non-kinetochore microtubules elongate the cell. 284 Chapter 11, Meiosis and Sexual Reproduction. Access for free at openstax.org. Figure 11.5. The process of chromosome alignment differs between meiosis I and meiosis II. In prometaphase I, microtubules attach to the fused kinetochores of homologous chromosomes, and the homologous chromosomes are arranged at the midline of the cell, the metaphase plate. In metaphase I and anaphase I, the homologous chromosomes separate. In prometaphase II, microtubules attach to the kinetochores of sister chromatids, and the sister chromatids are arranged at the midpoint of the cells in metaphase II. In anaphase II, the sister chromatids separate. Telophase II and cytokinesis, the chromosomes arrive at opposite poles and begin to decondense. Nuclear envelopes form around the chromosomes. If the parent cell was diploid, as is most commonly the case, then cytokinesis now separates the two cells into 4 unique haploid cells. The cells produced are genetically unique because of the random assortment of paternal and maternal homologs and because of the recombination of maternal and paternal segments of chromosomes, with their sets of genes, that occurs during crossover. The entire process of meiosis is outlined in Figure 11.6. 11.1. The process of meiosis 285. Figure 11.6. An animal cell with a diploid number of 4, 2n equals 4, proceeds through the stages of meiosis to form 4 haploid daughter cells. Comparing meiosis and mitosis. Mitosis and meiosis are both forms of division of the nucleus in eukaryotic cells. They share some similarities, but also exhibit a number of important and distinct differences that lead to very different outcomes. Figure 11.7. Mitosis is a single nuclear division that results in 2 nuclei that are usually partitioned into 2 new cells. The nuclei resulting from a meiotic division are genetically identical to the original nucleus. They have the same number of sets of chromosomes, 1 set in the case of haploid cells and 2 sets in the case of diploid cells. In contrast, meiosis consists of 2 nuclear divisions resulting in 4 nuclei that are usually partitioned into 4 new, genetically distinct cells. The 4 nuclei produced during meiosis are not genetically identical, and they contain 1 chromosome set only. This is half the number of chromosome sets in the original cell, which is 286. Chapter 11. Meiosis and Sexual Reproduction. Access for free at openstax.org. Diploid. The main differences between mitosis and meiosis occur in meiosis I, which is a very different nuclear division than mitosis. In meiosis I, the homologous chromosome pairs physically meet and are bound together with the synaptonemal complex. Following this, the chromosomes develop chiasmata and undergo crossover between non-sister chromatids. In the end, the chromosomes line up along the metaphase plate as tetrads, with kinetochore fibers from opposite spindle poles attached to each kinetochore of a homolog to form a tetrad. All of these events occur only in meiosis I when the chiasmata resolve and the tetrad is broken up with the homologs moving to one pole or another. The ploidy level, the number of sets of chromosomes in each future nucleus, has been reduced from 2 to 1. For this reason, meiosis I is referred to as a reductional division. There is no such reduction in ploidy level during mitosis. Meiosis II is analogous to a meiotic division. In this case, the duplicated chromosomes, only 1 set of them, line up on the metaphase plate with divided kinetochores attached to kinetochore fibers from opposite poles. During anaphase II, as in meiotic anaphase, the kinetochores divide and one sister chromatid, now referred to as a chromosome, is pulled to one pole while the other sister chromatid is pulled to the other pole. If it were not for the fact that there had been crossover, the 2 products of each individual meiosis II division would be identical, as in mitosis. Instead, they are different because there has always been at least 1 crossover per chromosome. Meiosis II is not a reduction division because although there are fewer copies of the genome in the resulting cells, there is still 1 set of chromosomes, as there was at the end of meiosis I. Figure 11.7. Meiosis and mitosis are both preceded by 1 cycle of DNA replication. However, meiosis includes 2 nuclear divisions. The 4 daughter cells resulting from meiosis are haploid and genetically distinct. The daughter cells resulting from mitosis are diploid and 11.1. The process of meiosis 287 identical to the parent cell. Evolution connection. The mystery of the evolution of meiosis. Some characteristics of organisms are so widespread and fundamental that it is sometimes difficult to remember that they evolved like other simple traits. Meiosis is such an extraordinarily complex series of cellular events that biologists have had trouble testing hypotheses concerning how it may have evolved. Although meiosis is inextricably entwined with sexual reproduction and its advantages and disadvantages, it is important to separate the questions of the evolution of meiosis and the evolution of sex, because early meiosis may have been advantageous for different reasons than it is now. Thinking outside the box and imagining what the early benefits from meiosis might have been is one approach to uncovering how it may have evolved. Meiosis and mitosis share obvious cellular processes, and it makes sense that meiosis evolved from mitosis. The difficulty lies in the clear differences between meiosis I and mitosis. Adam Wilkins and Robin Holliday I summarized the unique events that needed to occur for the evolution of meiosis from mitosis. These steps are homologous chromosome pairing and synapsis, crossover exchanges, sister chromatids remaining attached during anaphase, and suppression of DNA replication in interphase. They argue that the first step is the hardest and most important and that understanding how it evolved would make the evolutionary process clearer. They suggest genetic experiments that might shed light on the evolution of synapsis. There are other approaches to understanding the evolution of meiosis in progress. Different forms of meiosis exist in single-celled protists. Some appear to be simpler or more primitive forms of meiosis. Comparing the meiotic divisions of different protists may shed light on the evolution of meiosis. Merrilee Ramesh and colleagues too compared the genes involved in meiosis in protists to understand when and where meiosis might have evolved. Although research is still ongoing, recent scholarship into meiosis in protists suggests that some aspects of meiosis may have evolved later than others. This kind of genetic comparison can tell us what aspects of meiosis are the oldest and what cellular processes they may have borrowed from in earlier cells. Link to learning. Click through the steps of this interactive animation to compare the meiotic process of cell division to that of mitosis at how cells divide. http://openstax.org/l/how_cells_divide.